Hello everyone. Hope everyone is staying safe and healthy in quarantine. My journal club presentation today is on a 2019 article centered on organelle-specific targeting of polymorphisms into the cell nucleus. To begin to understand this article, the first thing we have to discuss is why the research is important. Nucleocytoplasmic transport is the transport of therapeutic agents across the nuclear envelope into the cell nucleus. This is a highly sought-after treatment in terms of gene therapy. Gene therapy is defined as the transplantation of normal genes into cells in place of missing or defective ones in order to correct genetic disorders. There is a long list of genetic disorders that nucleocytoplasmic transport could help treat, including but not limited to cystic fibrosis, Down syndrome, and hemophilia, where the blood does not clot properly. The problem with developing mass amounts of treatments for genetic diseases is the specificity of the nuclear pore complexes, which are the channels that regulate what is allowed to pass into the nucleus. Nuclear pore complexes regulate what is allowed in the nucleus because you don't want anything to be able to reach the genetic code and cause mutations that have disastrous effects on the cell. The genetic code has to be protected because when the cell divides and creates two daughter cells in mitosis, the DNA is replicated and each daughter cell is given an identical copy of the mutated DNA. These cells then go through mitosis and pass on their mutated DNA until eventually a large mass of cells have the mutated DNA, which can be detrimental to the host. The nuclear pore complex is a channel with a diameter of roughly 60 nanometers. Phenylalanine glycine nucleoporins, called FG-NUPs, are highly dynamic, intrinsically disordered proteins that are located within the nuclear pore complex central channel. To get access to the channel, cargo has to have soluble transport receptors known as carioferins, also called CAPs. The CAPs bring the cargo containing nuclear localization signals, referred to as NLS, into the nucleus through binding multiple times with the FG-NUPs. In order for the cargo being brought into the nucleus to be released, a small RAN GTPase binds to the carioferins in its GTP form. This releases both the carioferins and the cargo once inside the nucleus. Since this process is such highly regulated, there are many roadblocks in developing gene therapies. Some of these challenges include biocompatibility, biological stability, degradation, encapsulation efficiency, detection, and targeting accuracy. There has been some progress made, such as the use of using conjugated viruses like HIV, conjugating it to different types of nanoparticles. Some of the major unknown facts about nucleocytoplasmic transport is how nanoparticles that are larger than the channel diameter can still get through to the nucleus, and the mechanism of how synthetic compounds can bypass the nuclear pore complex barrier. This research study focuses on solving some of these unknown issues. The main goal of this journal article was to determine the nuclear targeting mechanism of NLS conjugated polymorphisms through the use of biophysical, ultrastructural, and cellular studies. They did this through developing biocompatible polymer vesicles that can pass through nuclear pore complexes. The purpose of this research is to develop synthetic nanomaterials that can shuttle therapeutic agents directly into the nucleus of the cell. The first step in this experiment was the formation of the polymer vesicles. The design and characterization of the nanocarrier is shown in Figure 1a. There were two polymorphisms synthesized that self-assembled into the nanocarrier. One was with the hydrophilic triblock variant called poly-2-methyl-2-oxazoline, block poly-dimethylsiloxane, block poly-2-methyl-2-oxazoline. And the other was a maleimide-terminated derivative of the same triblock polymer. The self-assembled nanocarrier was conjugated with a nuclear localization signal called bipartite nucleoplasmin through a catalyst-free thiolene click reaction. A control nanocarrier was also created through using the same copolymers, but it was not conjugated with the nuclear locating signal. These were referred to as the blank nanocarriers. Both types of nanocarriers were made to be compatible with the size of the nuclear pore complex channel. The cargo inside the nanocarrier was ruthenium red, referred to as RR, and Baudipy 630-650. This was incorporated into the membrane, as shown in Figure 1A. Both ruthenium red and Baudipy was used to test for nuclear delivery. Figure 1B shows the nuclear transport mechanism depends on karyopharins, specifically cap alpha and cap beta 1, to authenticate the nuclear locating signals on the nanocarrier. The karyopharins then bind to the FG nups and release the nanoparticle once the RAN GTP binds inside the nucleus. It can also be seen in the picture behind Figure A and B that the blank nanocarriers were not able to pass through the nuclear pore complexes due to their lack of nuclear locating signals. The next step was analyzing the characterization of the nanocarrier. In Figure 2A, cryo-electron microscopy was used to show both the blank on the right and nuclear locating signal tag nanocarriers on the left. Cryo-electron microscopy is used to show high-resolution structural images, as does transmission electron microscopy. The difference between the two is that cryo-electron microscopy uses frozen samples and a more gentle electron beam to prevent the destruction of more sensitive biomolecules. The cryo-electron microscopy showed both nanocarriers were spherical with hollow lumens. It was also determined that the membrane enclosing the lumen was thicker for the nanocarrier that contained nuclear targeting signals than the membrane of the blank. Figure 2B and 2C both showed transmission electron microscopy results for the size of both types of nanocarriers. In Figure 2B shows the nanocarriers charged with nuclear locating signals had a smaller general size than the blank nanocarriers. Figure 2C compares the radius size of the nuclear locating single-tag nanocarriers in red to the blank nanocarriers in black. This data was compared to the size determination by dynamic light scattering analysis, also called DLS, shown in the inset graph. The transmission electron microscopy was shown to have consistent results with the DLS in terms of the blank having a larger average radius, as shown by the graphs having similar peaks and differences between the two nanocarriers. In Figure 2D, dual color fluorescence lifetime cross-correlation spectroscopy was used to determine the amount of Baudipy and ruthenium red that was incorporated into the nanocarrier. The red line indicates the amount of ruthenium red located inside the lumen of the nuclear locating signal tag nanocarriers. The blue line showed the amount of Baudipy that was inserted inside the membrane of the nanocarriers. The black line indicates the cross-correlation of both ruthenium red and Baudipy located inside the nanocarrier. This data is then compared to the data of the inset graph, showing the amount of encapsulated and free ruthenium red in the red and green lines respectively. This data confirms that there is simultaneously encapsulation of both model cargoes inside the nanocarrier, as shown by the large cross-correlation amplitude and the shift of the individual cargo encapsulation towards longer diffusion times when compared to the free ruthenium red. Next, the scientists wanted to examine the binding affinity of the karyopharins to the nuclear locating signals of the nanocarriers. The karyopharins studied in this experiment where the heterodimer formed between cap alpha and cap beta 1. To study the binding interaction between karyopharins and the nuclear locating signals, dual color fluorescence lifetime cross-correlation spectroscopy was used again. In Figure 3A, increasing concentrations of nanocarriers were titrated against 20 nanomolar of cap beta 1 and 200 nanomolar of cap alpha. The signal cross-correlation measured on the y-axis increased as the nanocarriers were titrated due to the increased availability of the nuclear locating signal binding sites, causing an increase in binding. The picture in the top right corner of Figure 3A shows the specific binding of the karyopharins to the nanocarrier. In Figure 3B, the black squares show the binding curve of the karyopharins to the nuclear locating signals. The binding curve shows there is a maximum of 57 carrier per nanocarrier as shown by the leveling off of the graph. Once the carrier bind to the nanocarrier, the amount of free carrier complex decreases. The blue circles in figure 3b shows that as the number of nuclear locating signal binding sites increase, the concentration of free carrier decrease. Figure 3c serves as an analysis of the binding affinity of the carrier to the control nanocarrier. It is shown that the blank nanocarriers are not recognized by the carrier because there are no signal cross-correlation indicated by the black line. The image in the top right corner of figure 3c serves to illustrate how the carrier cannot bind to the nanoparticles and bring them into the nucleus. This provides a level of specificity as to what is allowed to pass through the nuclear pore complex. The next interaction studied was the binding of the nanocarrier to the FG nups located within the nuclear pore complex channel. In figure 4, the binding affinity of the carrier bound nuclear locating signals conjugated nanocarriers with the FG nups. In particular, the FG nups CNUP 153, CNUP 214, and CNUP 98 were analyzed. For all three of the graphs, the red line indicated the nanocarriers conjugated with nuclear locating signals. The black line indicated the blank nanocarriers, and the yellow line is for free carrier as shown in figures 4a, b, and c, respectively. It was found that the binding affinity of the NLS nanocarrier to each of the three FG nups did not differ from the binding affinity of the free carrier. The article explains this is likely due to the free carrier that are generally present when the binding of carrier with the NLS nanocarrier is at equilibrium. The control blank nanocarrier mixed with carrier showed a similar binding response to the standalone carrier. This indicates a lack of nanocarrier binding to the FG nups. It was also shown the blank nanocarriers and NLS nanocarriers both showed no FG nut binding in the absence of carrier meaning the carrier mediate the selective binding of the NLS nanocarriers to the FG nups. The article states that this selective binding is a prerequisite to bypass the nuclear pore complex selective barrier. The equilibrium dissociation constants and maximum response signals are shown by the inset graphs. Next, the researchers wanted to focus on the role of RAN-GTP in the nuclear uptake. In Figure 5, a permeabilized cell assay was done using HeLa cells and a mixture of RAN-GTP, key transport factors, and an energy regenerating system that reactivates the enzyme RAN-GEF, which converts RAN-GTP to RAN-GDP to RAN-GTP in the nucleus. The purpose was to determine if RAN-GTP promoted the nuclear uptake of nuclear locating signal conjugated nanocarriers. The scientists varied the amount of RAN-GTP in the mixture, as shown by the three rows in Figure 5a. The concentration of NLS nanocarriers was kept constant. As shown in the figure, there were negligible amounts of ODP-labeled NLS carriers detected in the nucleus after 2-hour incubation, when there was no RAN-GTP present. NLS nuclear imports was shown to be highest when the concentration of RAN-DP was the highest, as shown in the first column of Figure 5a. The signal of cap beta 1 was shown at all three concentrations of RAN-GTP, which indicates the carrier are necessary but not enough to cause nuclear uptake by themselves. It was then determined that nuclear uptake required the binding of RAN-GTP to release the carrier in order to release the NLS nanocarrier from the nucleus pore complex channel. DAPI was used for nuclear staining, as shown in the third column. The fourth column shows the merged signals of the NLS carriers, the carrier and the DAPI nucleus. Figure 5b shows the time-lapse imaging of the nuclear import rates of NLS nanocarriers and varying concentrations of RAN-GTP. The results showed the import rate increased and the concentration of RAN-GTP increased. Near physiological conditions of 5 micromolar RAN-GTP, NLS nanocarrier uptake was increased, but at 20 micromolar concentration, the uptake was increased significantly. The scientists then wanted to study how the uptake of nanocarriers was affected in live cells. Live HeLa cells were used to study the nuclear uptake of BOJB NLS characters and Nile red labeled blank nanocarriers into the live cells through time-lapse fluorescence microscopy. This is shown in both Figure 6A and 6B. In Figure A, the first column shows the stained nanocarriers. Host was used to stain the cell nuclei as shown in the second column of Figure A. The third column shows the merged imaging of the nanocarriers and the cell nucleus. It is important to show that the yellow blank nanocarriers were left outside the nucleus, while the red NLS nanocarriers were able to enter the nucleus, as shown in the merge imaging. Figure B shows the imaging of the NLS and blank nanocarriers along with the host-stained nucleus in the same cells. The differences in cellular uptake between the blank and NLS nanoparticles were shown to be the same as the differences shown in Figure A. Figure C is the time-lapse imaging for a 12-hour period for the uptake of blank and NLS nanocarriers. As shown by the increased nuclear intensity of the NLS nanocarriers, the nuclear uptake of the NLS nanocarriers doubled after 12 hours and was consistently higher than the uptake of the blank nanocarriers. The low increase of nuclear intensity of the blank nanocarriers can be explained by passive diffusion. Figure D compares the cell viability of the HeLa cells for 48 hours at increasing concentrations of blank and NLS nanocarriers, compared to a control of HeLa cells exposed to no nanocarriers. As shown by the bar graph, the HeLa cells remained viable when treated with both types of nanocarriers at all concentrations. The only significant decrease in viability was a very small decrease when exposed to 1.5 nanomolar of NLS nanocarriers. In Figure E, transmission electron microscopy was used to verify the individual NLS nanocarriers were transporting through the nucleus pore complexes, and if the nanocarriers were maintaining their spherical shape after they entered the nucleus. In the figure, the black arrows point to the NLS nanocarriers that have crossed the membrane at the point labeled NPC for nuclear pore complex, and crossed into the nucleus indicated by the letter N. The cytoplasm is labeled with the letter C. In Figure F, the statistical comparison of the NLS nanocarrier's location in comparison to the blank nanocarriers is shown. The results shown for the NLS nanocarriers were 30% were in the cytoplasm, 25% were in the nuclear pore complex channel, and 45% were in the nucleus. In comparison, 80% of the blank carriers were found in the cytoplasm, 5% in the nuclear pore complex channel, and 15% in the nucleus. The localization of the NLS nanocarriers was found to be regardless of size, with the nanocarriers being 69 nanometers in diameter, the same as 70% of the blank nanocarriers found in the cytoplasm. It was then concluded that the blank nanocarriers were not excluded from the nucleus based on the size. Throughout this research study, polymerosome nanocarriers were developed and their transport into the nucleus was mapped. Multiple findings have come from this research, such as carrier-afferents authenticating and binding nuclear locating signal conjugated nanocarriers. Multiple carrier-afferents copies per NLS nanocarrier is needed to bind to FG nubs in the nuclear pore complex channel, and RAN-GTP is needed to release NLS nanocarriers from the nuclear pore channel through binding to carrier-afferents. The next possible step for this research could include making adjustments to the NLS nanocarrier in terms of size, degree of NLS functionalization, and/or membrane thickness. Through testing these adjustments to try and optimize the amount of nuclear uptake, a better gene therapy treatment could be developed. Here are my references for this presentation. Thank you for your time.